Hi, everybody. We're going to talk a little bit about cells here. This PowerPoint will be shared with you in Google Classroom because I'm not going to spend a lot of time going over every individual cell organelle. I want to talk more generally about cells in this presentation. And so we're going to talk about cell types, cell theory, stuff like that. And then I want to talk to you about how we categorize organelles, but then I'm going to leave the looking up what each organelle does to you. And there are some organelles that we don't necessarily focus on in baby bio in this PowerPoint as well. So just look for that PowerPoint to be made available to you in our Google Classroom. So let's start with cell theory. So in baby bio, if you took the 9th grade course, we went through this. If you didn't, you may not have touched on cell theory. It's not something that's part of our curriculum in the general bio course. But the cell theory is a set of 3 statements that summarize our understanding of cells. And our understanding goes like this. First of all things are composed of at least one cell. It may be that you have a unicellular organism that's only one cell, or you can have an organism that's multicellular. The cell is the basic unit of organization of organisms, which means essentially the smallest component of living things that itself is still alive, we would call a cell. Okay, so organelles don't live, but cells do. And really what that means is that the cell can carry out all of its own life functions all on its own. And then finally, cells come from pre-existing cells. So cells don't poof into existence. They have to come from other cells that were there first that reproduce. For the moment, I'm going to skip over this Miller-Urey and go over that with you later on. We're going to do a presentation later on in this unit where we'll talk about Miller-Urey and endosymbiosis and things like that. All right, so PowerPoint's going to have some specifics on individual plant cells, right? And this is just a generalized picture of a plant cell. You can find a million of them on the internet, right? Here's a generalized picture of an animal cell. And obviously there are differences, and you're going to be pointing them out in the coursework that you're going to do. All right. But what you should remember is that because cells are small, that until the advent of the electron microscope, the cell was called the black box. We could see using a light microscope a cell. And we could see that it had a nucleus, right, if it was a eukaryotic cell, which we'll get to in a sec. And we knew that there was DNA in the nucleus, but we didn't know about all the other organelles that we now know about because there wasn't a powerful enough microscope invented to actually show us those organelles, okay? So a lot of the information that we have on the cell organelles comes from electron microscope analysis, but also scientists did a lot of chemical analysis. They did something called cell fractionation to give us at least some ideas of the chemical makeup of cells, where they would they'd freeze cells and break them up and then centrifuge the parts and take off pieces of what they got and do chemical analysis. So the human eye can't detect things smaller than 0.1 millimeters, and notice that eukaryotic cells tend to range much smaller than that. They range in the 10 to 100 micrometer range. 100 micrometers would be a fairly large cell. Prokaryotic cells are going to run from 1 to 10 micrometers, so they're going to be smaller. And then some of the smallest prokaryotic cells are almost 100 nanometers, which is even smaller still. Okay, and most of the cell's organelles are about the size of bacteria, but ribosomes, those are by far the smallest organelle that I can think of, are the teeniest. And in baby bio, we showed you this link. It's got a little slidey thing on the bottom, and as you slide it, it zooms in. Feel free to look that up. It's pretty cool, and you can get an idea of comparing different kinds of cells and how big they are, and to like a carbon atom, a water molecule, how big is a virus compared to that, and so on. All right, so we have 2 main cell types, and you may remember this from baby bio. You've got prokaryotes. Remember that pro means before, karyon refers to the nucleus. So these are cells that are the before nucleus. In other words, these are cells that are the more primitive cells. They lack a nucleus. They also lack membrane-bound organelles, which was a surprise until electron microscopes came along. Again, until we knew that those organelles existed, we didn't really know that other than the nucleus there was a difference. But then it makes sense that the prokaryotic cells would be smaller, right? Because they've got less stuff inside, so they're just a smaller, more compact cell. They do have a few organelles, though. All the cells, even though they don't have membrane-bound organelles within them, all cells do have a membrane, okay, including prokaryotes. So they all have a cell membrane that goes around the outside of the cell. It's just they don't have any internal membranes inside of the cell. They have a region called the nucleoid region, and it's a place in the cell where the DNA kind of clumps together. And the DNA actually kind of attaches to the side of the cell, the inside of the membrane in that nucleoid region. And there's cytoplasm and there's ribosomes, and that's about it. There's not really much inside of a prokaryote. There are two domains of organisms that are prokaryotic, and both of them are bacterial. So the archaea, domain archaea and domain bacteria are both consist of prokaryotic cells. Everything else is eukaryotic. So from the tiniest algae in the ocean to the largest elephant, okay, or a big giant sequoia tree. All other cells, all other living things are called eukaryotes, and that's because they're going to have a nucleus and then this very large array of membrane-bound organelles, as well as the full complement of regular organelles that are not membrane-bound, okay? And that means then that they have an outer membrane, but they've got these networks of membranes also within the cell. And what that's going to lead to is something called compartmentalization, which we're going to deal with also a little later on this week. But essentially, the cell can be kind of divided up into areas, and you can divide jobs to different areas within the cell. And that kind of helps the organelles to specialize and help them to function together a little bit better. All right, so here's your typical prokaryote. There's E. coli. And this is what it looks like with an artist drawing. This is what the electron micrograph looks like of E. coli bacteria. You have these fimbriae. Fimbriae are little extensions of the cell that can allow the bacteria to latch onto surfaces. You've got the nucleoid region here. It's the nucleoid. And what you can't see is that attached to the side, this chromosome that you see in the nucleoid region is actually attached to the membrane. You do have some ribosomes, plasma membrane, cell wall, and then some bacteria also have an outer. It's not a membrane, but an outer coating called a capsule that's tougher even still than a cell wall. And by the way, the cell wall here is different than a plant cell wall. Bacterial cell walls are made of different material than what you find in a plant cell wall. Plant cell walls almost exclusively are made of cellulose. Bacterial cell walls can be made from other materials. So this is different a little bit from a plant in that you're going to see it constructed of something else. And then, of course, here's your generalized structure of the eukaryotic cell, right? This one happens to be an animal cell. Now, rarely are you going to find all these things in the same cell. You've got microvilli here, which are little folds or extensions from the cell. You've got a flagellum here. You're not usually going to find those things on the same cell, but this is just kind of giving you an idea of what kinds of things, the variety of things that you can find inside of cells. So what we need to talk about now is categories of organelles inside of cells. And so essentially it's helpful to think of this as you have organelles at the top, and then there's two categories of organelles, those which are membrane bound and then those which are not membrane bound. And the not membrane bound would be things like the ribosome, right? That's a non-membrane bound organelle. And then within that membrane bound over here, that gets further divided because you have independent membrane bound organelles that function independently. And then you have these membrane bound organelles that are part of a system known as the endomembrane system. Okay. So the nucleus here is its own separate independent functioning organelle, but notice it's surrounded by a veritable network of membrane-bound structures here. And these membrane-bound structures that you're seeing are the endomembrane system. So you've got these foldy-uppy membranes that are extensively folded and surround the nucleus. This one is the ER, the endoplasmic reticulum. Here's the Golgi apparatus. These are all part of the endomembrane system. And what you can see is that kind of separates off this part of the cell from, say, some of these other membrane-bound structures like peroxisomes, mitochondria. If this were a plant cell, you'd see chloroplasts are membrane-bound, and so on. Those are the independent membrane-bound organelles. They're not in any way connected either by structure or function to the endomembrane system. Okay, so nucleus, we're going to skip over. You've got some information on the nucleus here. I'm just going to kind of quickly click. All right, so there's the nucleus and it's got its own nuclear envelope, which is its own membrane structure. It's a membrane bound organelle, the nucleus is in and of itself. And then again, that nucleus is surrounded by the endomembrane system. Okay, so you've got the endoplasmic reticulum, and notice that actually there's more membrane within the cell than you see surrounding the cell in most cases, right? Half of the total membrane of the cell is inside of this ER. Okay, that's how extensively folded that membrane is, just to allow it to get packed into the cell. You have two types of ER. You've got the smooth ER and the rough ER. The smooth ER has no bound ribosomes. Remember, ribosomes make proteins, right? So the rough ER that has bound ribosomes is generally going to be used for making and packaging and modifying and getting ready to send out proteins, whereas the smooth ER is going to be involved in the synthesis of other things, other non-protein components. It also can help with metabolism of glucose, detoxification, like converting toxins from the cell into harmless chemicals and things like that. All right, so there's the ER. That's part of the endomembrane system. And then you have the Golgi apparatus. So where the ER is more of a transport system that moves things around the cells, moves materials and is involved in the production of, but then also the movement of materials around cells. The Golgi is more of a warehouse and packaging area of the cell. It does some synthesis as well, but essentially the reason why this forms a system, even though these are two separate organelles, is that little tiny packages from one organelle, from the ER, they're called vesicles, can actually transfer themselves over to the Golgi, fuse with the Golgi, and then pass materials on to the Golgi. So the Golgi's function is sort of articulated with the ER's function. Okay, and that's what you're seeing here. Any membrane-bound organelle can actually pinch off a little vesicle, and this is a vesicle that's coming from the ER. And notice the ER, it's a series of flattened membrane discs, basically membrane sacs, called cisternae. Its receiving side is called the cis face. And so these little vesicles will come in, they'll fuse to the surface, they'll fuse to the surface of the cisternae, and then the contents of this vesicle empty into the Golgi where they're modified. And then notice once they get packaged, they kind of accumulate at the end of the cisternae, and add the more that you get actually removed from the cister, or the more of the materials that are ready to leave the cisternae, the more you get a more rounded, and then finally pinched off vesicle from the Golgi. And this can travel to whatever part of the cell that needs it, or it can even travel to the regular membrane, the plasma membrane, the cell membrane, where it can dump its contents out. All right, so you have lysosomes and vacuoles that are also connected to this endomembrane system. So lysosomes are storage sacs. And they contain really powerful enzymes. Some of them are powerful enough to digest cell parts, which it makes sense that you'd want to isolate those and keep them in their own little safe package away from functioning cell parts, so that, essentially the idea here is you want to digest only old, worn out cell components, and you don't want to digest the rest of the cell. All right. And then, yeah. And then there's some information here about some diseases of certain organelles, and that would be useful to you for your activity that you're going to do this week. Vacuoles are just large membrane packages for storage, general storage. And in baby bio, a lot of students think that only plant cells have vacuoles. That's not true. Animal cells do have vacuoles. It's just in plant cells, that large central vacuole is the most prominent organelle that you see in a plant cell. But animal cells do have vacuoles too. They're just usually a lot smaller and they're not as prominent, but essentially these are storage. And, the reason why these are connected to the membrane system is they can be on the receiving end of these vesicles from the ER and the Golgi as well. Okay, so both the lysosomes and the vacuoles can be on the receiving end. Okay, so there's information there about vacuoles. There is a specialized vacuole called the contractile vacuole. Remember, in baby bio, you should have seen a picture of an amoeba or a paramecium. And essentially, they have these little star-shaped structures. There's these little tubes that empty into a central contractile vacuole. So those tubes shove water into the central vacuole. The vacuole opens up. And then when it's full, it can actually pump excess water out of the cell, and it helps the cell to regulate its water. All right, so that's what these guys do. So there's pictures here. And again, you're going to have this PowerPoint available to you. So this is showing you how the lysosome can fuse with food vacuoles to help with digestion. Here, the lysosome is actually fusing with a vesicle that contains some old, worn-out organelles and digesting them. That's what you're seeing there. Okay, and there's what a large central vacuole looks like. All right, and then you have other organelles that are not part of the membrane system, but some of them are membrane bound. And again, ribosomes, not membrane bound, so not part of the endomembrane system, right? And then you've got these energy organelles, the mitochondria, right? And the chloroplast. So these organelles are not part of the membrane or the endomembrane system, but they themselves are membrane bound. And these are special because these are the product of a process called endosymbiosis. And again, we'll get to that. You may remember it from baby bio, but we're going to go into a little bit more detail with some of our activities this week. Okay. You also have some other vesicle-like organelles, so small sac-like organelles, but they are not necessarily connected to the endomembrane system, such as peroxisomes. Peroxisomes contain an enzyme that can break down hydrogen peroxide. Sometimes during the course of your metabolism, you accidentally produce toxins like hydrogen peroxide, and so peroxisomes are waiting to break that down. They can also break down fatty acids and amino acids. And it makes sense, right? Remember from biochemistry, normally you do hydrolysis to make polymers, right, as part of your metabolism. Well, what if you, instead of taking an H off of one molecule and one monomer and an OH off of another, what if you actually took off two OHs and you would produce H2O2, which is hydrogen peroxide? And that's what I mean when I say you accidentally can make hydrogen peroxide, which is toxic to your cell. And so you need a way of dealing with that. Vesicles are distinguished by vacuoles, by the way, if you're trying to figure out what's the difference between a vacuole and a vesicle. Vacuoles tend to be for storage, and they tend to be permanent structures. Vesicles are not. They're usually pinched off from another membrane, and then they usually fuse to something else and disappear, so they're not permanent. Okay. You also have some organelles that are there for scaffolding, such as the cytoplasm, right? That's the jelly-like goo that everything is floating in the cell. And then there's also a network of actual scaffolding, protein scaffolding called the cytoskeleton, which is a network of proteins that kind of hold the cell up into its shape. And what scientists have found also is that sometimes proteins will attach to that cytoskeleton and they will carry vesicles with them and move along the cytoskeleton to transport vesicles to various parts of the cell. So they can kind of act like the little Disney monorail where the vesicle can attach to the protein and get slid along until it reaches its final destination. And then you have connected to the cytoskeleton something called the ECM or extracellular matrix. And these are outside of the cell but still connected to the cell. And these are proteins as well. And they're the same kinds of proteins. It's just a matter of them, not instead of being inside of the cell, they're outside of the cell. And here's a picture of what that looks like. And then, of course, finally, you've got some organelles for movement. Okay, cilia and flagella are what you're commonly going to find in human cells. Cilia tend to be shorter and they tend to be more numerous. Flagella tend to be longer and you have fewer of them, usually only one or two. Some cells, some eukaryotic cells, move using a pseudopod. All of this should be reviewed from baby biology.